Hello, everyone. My name is Sanaj Punan from the Desai Sethi Urology Institute at the University of Miami. I'd like to start by thanking Faye and the organizers for the opportunity to give this talk entitled Liquid Biomarkers and MRI in Prostate Cancer and Whether or Not They're Complementary. So my main disclosure is this talk is sponsored by Exosome Diagnostics, but my goal here, you'll see, is not really to speak about the Intelli test or any biomarker in specific, but rather to speak collectively of biomarkers as a whole and how they can integrate with MRI. And then the last part of the talk, I'm going to focus a little bit more on the Exosome Diagnostics platform for extracellular vesicle analysis and RNA-seq. And I just think there's a lot of very interesting work that's being done there that I'd like to share with you. So this is from their NCN Guidelines, and we can see here for men undergoing prostate cancer evaluation for an elevated PSA, we can consider multiparametric MRI if available, or various biomarkers that can improve the specificity of screening, because we know that PSA may be prostate cancer sensitive, but not specific. Now, I know this audience is very familiar with the various pros and cons for MRI versus biomarker, and I'm not going to go into this in any detail, but I don't think that this is a either or type of approach, and these two tests can be quite complementary. We have shown this and others have shown this. This is work looking at 1,000 men from 8 different institutions, and you can see that the BioRads and the 4K do appear to be quite complementary. As the 4K score increases in range, you see the detection of high-grade cancer increase in range. As the BioRads increases along the rows here, you can see the cancer detection increases. And when you look at the AUC, or the performance of these tests, you can see that 2 tests together outperform either of these tests on their own. Now, when we looked at various methods of putting these tests together, so let's say you combine it and do both tests in everybody, so you only don't biopsy someone if they have a low 4K, less than 7.5%, or their MRI was favorable, PIRADS 3 or negative, then you only miss about 1% of grade group 2 or higher prostate cancers, but you only avoid 16% of the biopsies you otherwise would have done. Now, if you try to sequence them, let's say start with a 4K score. If it's low, don't do anything. If it's high, get the biopsy. And in the middle, you may need the MRI to figure out whether you do the biopsy or not. Well, then you avoid about 40% of the biopsies you would have done and miss only 6% of high-grade cancers. Alternatively, if you start off with the MRI and do a biopsy for PIRADS 4 or 5, and only do a biopsy if the 4K score is above 20% for those men with a negative MRI or PIRADS 3, you get very similar results. About 40% of the biopsies would be avoided, and you miss about 9% of grade group 2 or higher cancers. So we did not see a big difference whether you start with an MRI or whether you start with a biomarker in terms of cancers missed or biopsies avoided. UCSF also did something very similar, looking at men who had a 4K score, an exosome test, or an MRI, and then underwent biopsy. And you can see a list of various algorithms or combinations of testing here. And on the left, they're looking at the percentage of biopsies avoided. On the right, they're looking at the percentage of high-grade cancers missed. And you can see a very obvious pattern here that the more biopsies avoided, the more high-grade cancers missed. There's no perfect test that's only going to avoid a biopsy in someone without a high-grade cancer. So this was just something that they saw. But they wanted to look a little further into this whole concept of sequencing and whether you started with a biomarker or started with a MRI. Now you can see the panels to the left here are speaking to starting with a biomarker. So if you look at the bottom, if you got an exosome and it was low, less than 15.6, you did nothing, you just observed the patient. If it was high, they considered it greater than 19, then you did the biopsy regardless of the MRI. And in the middle, you got the MRI to decide whether you needed the biopsy or not. Above here is a very similar algorithm using the 4K score instead of the exosome test. But in general, if you did this approach, you would avoid about 40% of the biopsies you otherwise would have done. You miss about 5% of grade group 2 or higher cancers, and you would have avoided about almost a third of MRIs. Now, alternatively, if you start with the MRI, if it's positive, PIRADS 3, 4, or 5, then you're doing the biopsy. If it's negative, PIRADS 1 or 2, then you get the exosome and use the exosome to decide on biopsy. And up here, they were replacing the exosome with the 4K. Now, what they found here is when you started with the MRI in such a fashion, you ended up avoiding only 17%, close to 20% of the biopsies you've done, compared to 40% if you started with a biomarker. And here, you can see, again, missed cancers is about 2% versus 5%. So you did find that in their study, starting with the MRI resulted in a lower number of biopsies avoided, but a lower number of cancers missed. But a lot of that could be the differences how, they looked at PIRADS-3 in this study versus ours. And also keep in mind that both of these were observational retrospective studies. A lot more prospective work needs to be done looking at the sequencing to decide, the difference and the impact of starting with an MRI versus starting with a biomarker. Now, I'm just going to switch gears a little bit to speak about MRI and talk a little bit about the work that we are doing at the University of Miami. My collaborators, Dr. Alan Pollack, who's giving some talks later on in the conference, and Dr. Radka Stoyanova are big believers in quantitative multiparametric MRI of the prostate. And Dr. Stoyanova has actually created this habitat risk scoring system, which is an algorithm that reads the prostate on a pixel by pixel level and actually creates a heat map assigning regions of risk. And when we've tested this in patients who had radical prostatectomy at our institution, we found that it did a better job than standard of care MRI in detecting volumes of Gleason grade group 2 or higher. So we were actually funded by the NCI to look at this prospectively in men undergoing evaluation for prostate cancer. So the premise is if you have a man who's referred for suspicion of prostate cancer, elevated PSA, and the endpoint would be a biopsy, currently we get an MRI, and that has about a negative predictive value of about 70% or so for predicting a grade group 2 or higher prostate cancer. Now, that means 30% uncertain, which is not exactly great. Now, we believe that adding our algorithm can take that uncertainty or the certainty to about 84%, and adding a biomarker brings it up to above 92%, giving a much more certain sense of whether the person does or does not have cancer. And if you can be 90% certain that you don't, because these tests came back negative, you may feel more safer avoiding that biopsy. Now, that's the hypothesis behind this trial. We have about 250 men that we're trying to accrue, and we're about a quarter of the way there. But we do send off a lot of correlative biomarkers. We're getting a 4K, we're getting an exosome, and again, we're getting an MRI read by standard of care PIRADS and this quantitative MRI. So there's a lot of information we're getting off this trial, and I hope we can learn a lot from it as well. Now, in the last part of this talk, I wanted to talk a little bit about exosomes and the Exosome DX platform. So for those of you who don't know, exosomes or extracellular vesicles are things that are secreted from every living cell to mainly communicate with the environment around them. But tumors, or localized tumors even, secrete them at about 20 to 30 times the rate. And these secretions carry with it RNA, DNA, lipid, protein, all the constituents of the parent tumor that are very well preserved in a lipid biomembrane. So unlike a lot of the advanced tumors, circulating tumor cells, first circulating free DNA, which is often dead kind of cells, these cells have their DNA and RNA very well preserved. And as a result, they can be a great source of various biomarkers. Now, the group at Exosome DX has a very robust and established pipeline for the isolation of extracellular vesicles from various biofluids. You can see they've done it in CSF, in plasma, serum, and urine, and they're able to actually extract RNA and perform RNA-seq on extracellular vesicles from any of these secretions. You can see they look at over tens of thousands of RNA targets, the majority of them being protein coding RNA targets here. We actually did a small pilot with them. This was done by my collaborator, Dr. Sandra Gaston, and she sent plasma samples from 9 patients on my active surveillance trial, 11 patients on Alan Pollack's high-risk radiotherapy trial, and 11 healthy controls. The plasma was sent to Exosome Diagnostics, who filtered out the extracellular vesicles, the isolated RNA from those vesicles, and then did RNA-seq. And you can see with some machine learning, sophisticated techniques, they were able to whittle down to 4 candidate genes that could reliably tell whether that plasma sample came from a high-risk patient or a low or benign patient. Now, obviously, this is a very small pilot, and we need to validate it. We are sending more samples from both of these trials to verify these candidate signatures and see if there's any other signatures that may add. But we're also looking at opportunities to validate other signatures, perhaps radiation response, androgen deprivation response, and other things as well. Other correlative work that we're doing with the company based off this MD Select trial, which I just showed you, looking at the quantitative MRI, is this whole concept of tissue-specific EV enrichment. So certain EVs are tagged with antigens that may tell us where that parent cell of origin from that EV could be. And if you can enrich for EVs that carry that antigen, then you're going to have a pool of EVs that are coming from a particular organ and provide more specific information about that organ. So for example, if we look at PSMA as a surface antigen that we expect to see on prostate cancer cells, we can actually isolate using these antibody coupled beads EVs that actually have PSMA surface antigen and enrich for them. And that should be giving a sample of EVs that are more likely to be coming from the prostate, more likely to be coming from the prostate cancer, and can tell us a lot more about the biology of that parent tumor just by looking at the EV that we're extracting from circulation. So this was some preliminary work that they've done looking at 18 different known prostate cancer genes that's listed from A to R here. And when you look at the expression of these genes compared in PSMA enriched EVs versus IgG EVs, you can see the yellow boxes, which are the PSMA enriched EVs, are showing a higher expression in almost every single one of these genes. So again, perhaps enriching for PSMA carrying antigens will allow a pool of EVs that will be much more specifically from the prostate, provide much more reliable prostate cancer risk assessment, and therefore a whole new platform for biomarkers to come. So you can see this is very exciting in terms of what they're capable of doing. And this is not just in prostate cancer. You could talk about almost anything where there could be a role for biomarkers. So to conclude, I hope I've shown you that MRI and biomarkers do appear to be complementary. They seem to work well together, but the right sequence still remains unclear. I hope I showed you that extracellular vesicles are exciting. They can be secreted by all local tumors and easily extracted, making a wonderful source for various biomarkers, and that the ExoDX platform is very robust and has significant potential for many of these indications going forward. I'd like to thank you again for your attention and happy to take any questions you may have.